Hi everyone. My name is Dongyan Lin. I am one of the program committee members of this year's CCN, and I have the pleasure of being the session chair of this KNT tutorial in silico neuroscience and emerging paradigm for brain discovery. And without further ado, I'm going to let Alessandro and Domenic take it away.
All right, cool. So, hello everyone, and welcome to our little tutorial session on in silico neuroscience, an emerging paradigm for brain discovery. So here you will find our QR code, which you can scan to reach our Google Colab tutorial. Question before: do you all have internet access on your computer? Does it all work out? Yes? No? Because same, I had the same issue earlier. um, maybe Aldo want to maybe help with the connection because we might have some issues with the internet. Um, oh. Yeah, because we didn't pop out either. All right, okay, but either way, of course, thank you all for coming. I'm very happy to see so many faces here that you guys are interested in joining our little tutorial. Again, of course, this is the QR code to our collab. If you cannot scan it with your laptop, which might be the case, you can just enter the tiny URL, which will also lead you to it. If you have any problems with this step, let us know, then we can try to help you with that. Um, then I think I would go ahead and start switching to the collab. Is anyone not ready yet for me to switch? All right, okay. Then, okay, so once you're here, you will see our little Google Colab tutorial. The first thing I want you to do is to change the runtime type because we will be doing some heavy computations and basically Google Colab starts with a CPU. So what I want you to do is to click on this little arrow here and change the runtime type to have a GPU ready because otherwise it will be super slow. Huh? Bigger? All right, that wasn't. Can you help me?
What have you done, man? What have you done?
Over there.
Yeah. Okay. Okay, I will check again.
Okay. Is it readable from the back? Okay, I got a shaking hand. Is it better? Perfect. All right, okay. So again, if you haven't done it already, please change the runtime type to have a GPU ready. And yeah, then once you are here, you will see the overview of our little toolbox. In the first section, we will have basically some introduction on the in silico neuroscience, basically the same things we talked about in our keynote. Then we have some information here about our toolbox BERG, which you can read if you want, and then our tutorial schedule, which we will follow now. So, like I mentioned in the keynote, we will follow the tutorial in 3 stages. In the first stage, we will introduce BERG to you, where you will also generate your first fMRI in silico neural responses, and then also visualize them. And the second stage will be hosted by Ale, and there you will learn how to validate your in silico neural responses and show that these in silico neural responses also capture fundamental organizing principles of the visual cortex, which will be category selectivity and retinotopy. And in stage 3, you will run your own experiments on neural control or on relational neural control, but more on that later. Like I said, stage 1 and 2 will be guided, and stage 3 will be on your own, but again, we will be here to assist you if you have any questions or something goes wrong or have a question or whatsoever. Also, each of the stages are able to run independently. So that means if you, for some reason, don't want to play with stage 2, you are still able to run stage 3 because you don't need to run all of them. The only thing you have to run is the stage 0, which will be all of the installations. That being said, we can go further down. Here you will find citations, also further resources like the website, our GitHub, our AWS buckets, and of course a link to our keynote and tutorial page. Okay, so if you guys are ready, I think I would go ahead and start with section 0, which is basically just the installations. If you haven't done it already, click this cell so you can install all of the libraries and all of the dependencies that we need. And once that is done, you can import all of that using this cell here. And next we have to access our tutorial data. And the thing is, usually BERG works with AWS, so all of our data is on AWS, but for the sake of the tutorial, we thought it might be easier to have a subsection of all of our available data on Google Colab, on Google Drive. And you now have to access it, and it's super easy. What you basically have to do is you have to open this link here, which will lead you to our, oh, okay, never mind. No. Sorry for that. You have to click on this, and this will lead you to our Google Drive folder. And what you have to do is you have to create a shortcut that will lead to your own drive. And you do it by doing the following. You click on this little arrow here, then click on Organize, then click on Add Shortcuts. And what will happen next, you can click on all locations and then on My Drive. And what will happen by doing that is you will basically just create a shortcut from your Google Drive to our folder, and thereby you have everything accessible while still loaded in our space. So once you have been doing that, you can go back to your wait, let me do it like this. So you can go back to your to the Google Colab, and everything should be connected. And now you have to mount your Google Drive to Google Colab. You just have to accept everything that it requests you. Ale, I need you. Sorry for that. Perfect. And click on continue. Okay, and once that is mounted, the next thing you have to do is you will have to select the directory where you have saved your the connection to your folder. If you didn't change anything and you saved it to your drive, you can just execute the cell because it will be correct already. And then in the next cell, we will initialize the PyCortex subjects. This is only for visualization of the responses across the cortical surfaces. And that concludes it. Here you will see an overview of our tutorial data. This is how the tutorial data is stored in AWS. You will basically have a folder with the encoding models, and for each of the modalities, we have a separate folder, and within each of those modality folders, for example, for fMRI, we have a separate folder for all of the data sets, for example, NSDFS average, and within those dataset folders, we have the model folders at the end, which will then include the model weight, the model accuracy, and the additional metadata. And again, since our tutorial will mainly focus on the fMRI models, only those models are included on Google Drive. All right, okay. That's the section 0, which was the installation. Now comes the fun part. Now we can finally start working with BERG. So the first thing you have to do if you want to work with BERG is you have to create an instance of BERG. And by doing that is you just use BERG, which you have imported earlier, and load it with the directory we have just declared. So it just basically loads up BERG and connects it to the folder so it can access all of the encoding models and the metadata and so on. And BERG has a variety of different encoding models, and all of those models are also on our website. But if you want to see them directly in the code, you can use the function berg.listModels. And what this function does is basically list all of the available models that we have, and they follow a specific pattern. They follow the pattern modality, which is fMRI. Then the data set, in this case, is BMD, and then the model is 3D. For example, we also have it here for FMI, and the CFS average, and the model then is AlexNet. We also have additional models. For example, we also have all of the brain score language and brain score vision models, but you have to also install a different version of BERG because there are some dependency issues sometimes. Same applies for the Tribe version 2 model. Yeah, this is, oh my God.
Do not touch this.
Okay, thank you. Sorry for that, it's not my computer. Then, if you also want to have a better overview on what datasets are available, you can also execute the berg.getModelCatalog function. And what this one does is it basically prints out all of the modalities that we have, so ECOG, EEG, and also tells you all of the datasets we have with this modality. So you have like a nice overview of what's possible, basically. And yeah, enough about that. Now comes finally the fun part. Now we can finally use BERG to generate our in silico fMRI responses. And for doing that, we first have to prepare the images we want to generate the responses to. And here in the first cell, what we're basically doing is we're going back to our BERG directory, which we have declared earlier. And here we have a folder where we have stored images. And what this cell is basically doing is it just gets all of the images, parses them, transforms them, and puts them into an array. And once this is done, you will see that we have an array of the shape any second. All right, you will see we have an array of the shape 100, which are the 100 images we have in the folder, and then, of course, the image dimensions here. And these are the images we are working with, and if you want to see how those images look like, you can go to the next cell and use the slider to see what each of those images look like. So, for example, if I want to see how image 34 looks like, I can execute this, and I can see it's a picture of a bridge, or if I want to execute it for this image number, which is 53, I can see a person on skis. All right. Then next now we have our images. Now we want to select a model with which we are going to compute the in silico fMRI responses, and we are selecting a model by Huse, who is the winner of the 2023 Argonauts Challenge. And this is the model ID, and the model ID you could see in the list model function we had earlier. And for every model that's available in BERG, we have a big model card available, which I can show you now. Yes. So again, for each of the models, we have a model card ready, which basically has more information on the model, for example, like the modalities, species stimuli, a thorough description for each of the models is also available. Then metadata, we also have additionally available, more on that later as well. Then also we have information on the input types for this model, because every model might have a different input, the output types, because these might differ as well across models, then the parameters for each of the functions we have in the model cards, and most importantly, we also have a code example for the example usage. So if you don't want to think too much about it, you can just go to the model card, copy and paste it, and just insert your images, and then it's super fast to generate in silico responses to that very model. So now, if you don't want to go onto the website, you can also use the berg.describe function. It basically does the same. It just prints everything into the terminal. It's just basically the same information we had earlier, including our little coding example here. Second. Okay, sorry for that. Okay, so the next cell will be the preparation of the encoding models, and in BERG, each subject has their own encoding model to predict in silico responses, because they are usually trained to their specific brain structure. And what we are using for that is the function getEncodingModel, which takes the model ID as input and the subject. And here you can select with a drop-down menu which subject you want to generate the in silico fMRI responses to. If you also want, you can also select the region of interest you want to select the brain responses to. I will select none, which will mean that we will generate the responses to the entire cortex. What you can do if you want, you can select a certain region of interest and then compare how your results will differ compared to what I'm producing here. So I will execute this cell now. And what it will basically do is it will just prepare the model into this little variable here, and load all of the data, prepare the weights and everything, so that we can use it in our next cell. Sorry for my scrolling. Okay, so in the next cell comes the most important part, the final encoding. So for that, we are using the function encode, which will take as input the encoding model we have just created, the images we put into an array earlier, and optionally, if you want, we can also set return metadata to true, which will basically just also return the metadata. Again, I will explain the metadata in the next second. And if we do that, it will generate the fMRI responses. Yes. And while this is computing, I can already go further. If we print out the shapes of the responses, we will see that for the left hemisphere, which is just the silico response which we just created, the first index, because we can see in the model card, if I scroll up again, the output will be left hemisphere vertices, and in the second index will be right hemisphere vertices. Again, for each model it's different, so always check out the model card. And if we execute this, we can see that is exactly that, so 100 images, and then we have all of the vertices for each of the hemisphere. All right, so now we are wondering, okay, we have 100 in silico fMRI responses. How do they actually look like? And what we can do now in the next cell is select an image and then visualize those in silico fMRI responses. So for me, I'm, for example, interested in image 74. Let's look at it. It's a person playing with a kite. And I want to know, okay, how do they look like projected to the brain? And what we're doing basically in the next cell is the following. If I haven't selected a region of interest, which was the case for me, we are basically going to the left hemisphere data and select a row which corresponds to our image number. We're doing the same for the right hemisphere. If we have selected a region of interest, we will also parse it with the RI indices, which are available in the metadata, which basically just like tells the data which indices of the array corresponds to a region of interest. And then in the next cell, we can use PyCortex to visualize it on a inflated cortical surface. And since it's a 3D image, sometimes it takes a few seconds to show up on Google Colab. Let's see. Okay, while this is loading, I can already mention that we also in the next cell, we have the same visualization for the cortical, there it is, never mind. All right, so here we can see how the predictions look like projected onto the brain. And if you have done it yourself with a region of interest, you should ideally only see like a section of it, so everything else should be gray, and only the region you have selected should be visible looking at the activations. And the next cell will be the same thing, just projected on a flattened cortical surface. And once this is done, oops, sorry, I'm super bad at scrolling today. We can see the same thing just projected on a flat map, and we can see how the brain responds to the image we have just looked at. All right, so that is that, and I've mentioned multiple times the metadata, and you're probably wondering, what am I talking about? So the metadata is, I mean, we have many information on our model cards already, and there's usually more information to that for each of the models. For example, each model might have information on where are the regions of interest, or like what are the encoding accuracies, or subject information, or noise ceiling signal to noise ratio. And these values we also provide for each of our models. And if you want to see the metadata for a model, what you can do is you can either use the function getModelMetadata, which is here, where you just enter the model ID and the subject of your choice. Or what you could also do, we saw this earlier when I was encoding the data, is just put return metadata equaling to true, and then you will also have the metadata returned there as well. And then we can look at the metadata. I will not execute this now. We can see basically the same thing we also saw in our model card earlier. So we have all of the values for left hemisphere, noise ceiling signal to noise ratio. We have all of the indices for each of the regions of interest for both hemispheres. And if we scroll further down, Oh my God. We can also see something like encoding accuracies, or also which numbers have been used for training images, which numbers have been used for validation images, and so on. So always like helpful information to understand what's going on in the model, how reliable is this model. And if you want to like look closer into it, we can also select a region of interest and print out the values of the metadata. I'm selecting now V1D. And we can see, in general, the NCSNR scores, they look like this, for example, for the first couple of indices. The region of interest, vertex indices for the selected ROI look like this. So again, these are just the indices. So 34 means the 34th index is region of interest V1D. And we can also see the NCSNR scores for that selected region of interest. Yeah, okay. I think that is it with my part on the introduction to BERG. Next comes stage 2. Yes, a question. So the idea of having different subjects is because we have one encoding model for each of the subjects, and each subject has their own view, basically. And we just want to make sure that we can, because, for example, for this case, we have all of the NST subjects, and we just don't have an ensemble model, if that's your question. We just thought it might be better to have like one model per each of the subjects. Yeah, of course. So, for example, we have the NSD fMRI responses, and we train the encoding model to predict those fMRI responses. And what we're doing with BERG is we show this encoding model that has been trained to predict the actual fMRI responses to the images the person actually has seen. And what we do with our encoding model is we have all of those weights now, so we put in a new image and then see, based on what the model has learned so far, how would it predict how the brain should respond to this image. Yes, exactly. Okay, want to take over?
Yeah, following up on the different subject. So why might we want different subjects for different models for each subject? One benefit or advantage of this that we will see in this stage 3 is that you can actually, when running experiments in silico, you can cross-validate your results across subjects. So are the findings that are the things that you find in specific subjects also generalized into other subjects, which already gives you a hint potentially that what you're finding is real and not some specific artifact of the model that you're using. But we will see this more in detail during the third stage. So then during the second stage, or better, like for now, the idea of the tutorial, the first stage is just to, maybe it was a bit fast to follow, but it's just, this is a bit just to give you an idea of what the toolbox consists of. It would be, yeah, later on, you can also check this later at home and go through the tutorial at your own pace. There's a lot of text, so hopefully you will understand what each step consists of. Now, like, what we would like you to get is more the intuitive meaning behind this, like the intuitive idea or general idea what the toolbox can do or cannot do. And during stage 3, you will run your own experiments, so that will be more hands-on. That being said, if you have questions, again, please just raise your hand at any point. So during the stage 2 of the tutorial, probably in this stage as well, I would, it probably is better if you just follow me instead of running the code yourself, which you can do at home. But it's more, for me, it would be more important that you get the point that is conveyed through the stage. So why is it important? So in this stage, the idea is that when you want to use encoding models to generate in silico responses, and then you want to use these in silico responses for experimentation, you want to make sure that these in silico responses are capturing the underlying brain phenomena, brain mechanisms that you actually are interested in studying. Because if they don't, then your in silico responses will be useless. And one way of checking that brain responses are actually capturing potentially things that you're interested in is looking at their prediction accuracy. So how similar are these predicted in silico responses to responses, corresponding responses from real brains? And to check this, typically the models coming with BERG, they also have metadata that Domenic was talking about, and part of this metadata is also the encoding accuracy of the models, which you can then plot. For example, here again we're focusing on encoding models trained on the NSD, and this is their encoding accuracy. This is just correlation between predicted and recorded and measured responses. You can see, okay, unsurprisingly, correlations are higher over visual cortex, but they're pretty high to reach also levels of 0.8. This is in distribution, but you might also want to check out of distribution accuracy. So this is encoding models evaluated on NSD synthetic, and you see that accuracy is lower, but it's still present, especially in visual cortex. So why is this important? Again, if you want to use these encoding models, you want to know that their predictions generalize to stimuli that you might be using for your experiments, but also importantly, you might want to know how good they are in generalizing out of distribution, because it might be the case that you want to run in silico experiments using stimuli that are quite different from the stimuli on which the models were trained. So you want to know whether these models generalize out of distribution. This is just an index that, to a certain extent, they do. They're not as good as when they're tested in distribution, but they're nevertheless generalizing. And below, I also give an example of like actual evidence that these models are good and they work when also tested out of distribution specific tests. So this is one way of checking the validity of the models. So you should always ask yourself, is the model that I'm using accurate enough? Is it predicting brain responses accurately such that I can rely on these responses and use them for in silico experimentation? That's one way of testing, looking at prediction accuracy. The other way that we also introduced and proposed during the keynote part is to check whether these models capture properties, organizing principles of, in this case, visual cortex, that we know to be true from previous literature. So this could be retinotopy, this could be category selectivity, but could be other things. And here we just show that, so this is just code to replicate the results that I was showing you earlier during the keynote. So during the retinotopy part, we want to check whether the in silico responses reproduce polar angle maps and eccentricity maps. So what we do more in detail is we pick one stimulus, so it could be any stimulus. This is the stimulus that we picked. And then we want to create many probe images for this specific stimulus. So probes that only show the portion of the stimulus under one specific x and y visual angle coordinate. So we want to get ultimately something like this. So you see here, these are the probe images. For each x and y visual coordinate, there is one specific probe. And we get all of these probe images and we feed them to the models. So here you see we're doing the same steps that Domenic was showing earlier. You initialize BERG, you load the encoding model of your choice, you select the subject, you select the model ID. Once you load the model, you use the loaded model to predict corresponding responses for these probe images. Then you get one response for each probe image. And then you convert these responses to polar angle and eccentricity maps. So this is really specific to PRF mapping. I don't want to get into the details. The more general idea is that, let's say that you're interested in some replication. You want to see whether these models capture some organizing principle of vision or also other stimulus modalities of your choice. You can test it by running the same experiment that has been performed in previous literature, for example, for face selectivity, for retinotopy, or other types of neural signatures of your choice. The only difference being that instead of using in vivo data, so measured responses, you generate in silico responses for those same images that were used during the article that you're interested in. So then once you do this, you get these maps. So this is the same results I was showing earlier. And similarly, we can also do this for category selectivity. This I was also showing earlier in the talk. The only difference here is the stimuli. Well, one difference is stimuli. So you have these images of faces, bodies, or food, or also houses. And one thing to notice is that these stimuli are, if you will, out of distribution with respect to the naturalistic stimuli on which the models were trained. Because again, the models were trained on natural images, color images, so the stimuli from NSD natural scenes data set, they come from the COCO database originally. Whereas these are single objects cropped grayscale on artificial background. We nevertheless ran the models on these stimuli to probe for category selectivity. And what we found is eventually, this is just the code to compute the t-contrast maps, but the t-contrast maps do replicate, do validate what we know from literature in the sense that like high values correspond also to regions correspond to the ROI labels, which were defined in vivo from the NSD experiment. So this is faces, so you see that you have higher T contrast values in face areas. This is bodies. And interestingly, you also find food selectivity. So there has been papers which have shown food selective areas, and they show them to specifically be adjacent to the FFA, in between FFA and PPA, which is also where we find the higher T values for food compared to other categories. So it's also replicating new category selectivity, in this case for food, that has been shown recently. And once again, and this is houses. And once again, this is all validated through stimuli which are out of distribution with respect to the stimuli with which the models were trained. Do you have any questions on this? Yes. Yeah. Yeah, so in EEG we have really the same rationale, only that the syntax will change in terms of the model ID. So in EEG we have models trained on things EEG2, which is this large-scale EEG data set of responses to images of objects from the things database. Over there we also have models trained on single subjects, so for each subject you will have an encoding model. And the syntax is just the same. You will have this get encoding model function. You will have to change the model ID to the EEG model of your choice. Select your subject, and you will get the model, and then you can encode responses for stimuli of your choice. Yeah. Other questions? Okay, so again, the important part of this, take-home message of this part is that whenever you use encoding models for, let's say, in silico experimentation, you need to be aware of what they can do or cannot do. I propose two ways in which you can have a reasonable estimate of whether they are suitable for what you're trying to do. So it's prediction accuracy of replication of known neural signatures, but you could also think of your own tests. And like importantly, if the models fail in showing the effect that you might be expecting when experimenting on silico, it could be that that the effect is actually not there in the brain. So it's like correct result. There you had an hypothesis, the model is testing that, and nothing shows up. So maybe your hypothesis was wrong. But it could also be that the models are just not capturing the underlying neural signature that you would like to test. And with that, I'd like to move to the third stage. That's the one actually interesting, when you will get to do something. So for this, it would be good if you actually, so for this, you will need your laptop, because I will not go through, like I will give a brief explanation, but the idea is that you then do your in silico experiments on your own. We will be walking around in case you need help. And afterwards, it would be nice to have some 10 to 20 minutes at the end, where you can also share what you found, what you didn't find, if there was any interesting observations, because you will be exploring selectivity across the entire cortex in different ways. So there's a lot of regions to choose among. So it would be also nice for us to hear whether you find something interesting, or if you have any ideas how to make things different, or how to improve the toolbox that you'll be using, etc. So for this, it is, again, it would be good if you all had access to, or at least the ones of you who wants to try it out, who have access to the tutorial. So if some of you are still having issues, or if you didn't quite catch the first configuration part, where you were importing the data, maybe you could raise your hand or let us know, and Domenic will come and help you, okay? Is there, okay, so you see some. So in the meanwhile, the point of this is to yeah, during this part, you will run some in silico experiments. It will be highly guided in the sense that we channeled what the purpose of the experiments will be, but it will also be highly interactive in that you'll get to make several choices, especially in terms of the areas that you want to test. So we divided this into 2 possible tracks. You can run one of them, you can run both of them. If you finish one, you can check out the other. So the first will be neural control. So neural control means that you will have all of these different areas in the cortex. So this is a parcellation for the HCP. Or alternatively, you also have ROIs from the natural scenes dataset, since the models you'll be using is from the natural scenes dataset. And the goal, the purpose of this track, of this is you pick one area of interest, and you want to find stimuli, images, that maximize responses for that specific area. So the idea is, what is the area tuned for? What is it tuned to? What is it functionally selected for? Which type of visual feature it is. And for this, you will be testing lots of images, tens of thousands of images. So you will run these tests across tens of thousands of images, naturalistic images, to find the subset of images that maximize responses in areas of your choice, okay? So across the entire brain. So this is the first track, whereas the second track is relational neural control. So I was talking about during the talk. Here the question is not what stimuli drive one area, but rather which stimuli align or disentangle representations of 2 areas. So what is it that areas encode similarly, and what is it that they encode differently? You might find some obvious examples. If you think about the FFA and the PPA, for example, in visual cortex, you will find that disentangling images are images that, for example, drive FFA, fusible face area, and suppress PPA. These will be faces, or the other way around, images that drive the PPA and suppress FFA will be images of places. Okay, that's intuitive, that's what you will find. But you could also ask more, let's say, interesting questions. For example, take 2 areas which have similar functional selectivity, such as PPA and OPA or RSC. So what are images that align them or disentangle them? And the images that do disentangle them, can you spot some visual properties of these images that lead to areas which are tuned to similar spatial layouts of the environment to actually differ? So you can really play around with this and ask different questions. So these are the 2 tracks. I'm going to give you a bit more information regarding 2 tracks that will that's useful before you jump into the code. So the first is the stimulus set. So we provide you with 2 stimulus sets, so 2 image sets, across which you can run these experiments, across which you can explore. So the first is, so this is wrong, let me just check, this is 50. Okay, so the first split is the 50,000 images from the ImageNet validation split. So you'll have the opportunity to run your experiments on these 50,000 images. So they come from ImageNet, so they are images of objects. Whereas the other split is, or other set of images you can use is 40,000 images from the MS-COCO test split. Okay? So these are not images of necessarily objects, but they are naturalistic scenes. So it could be that in each image has many different objects, or not even objects, but scenes. So these are the 2 image sets that you will be able to choose among. Again, there will be drop-downs for this. So it's you basically, the exploration part, the experimentation part of this stage consists of you making different choices through drop-down windows. Of course, the code is there, but you can check the code at home to know exactly how it works. But for now, the part we would like to do is more exploring in an easy user interface way. Another point is that for this part, for this stage, we did pre-generate the in silico fMRI responses for you. The reason being is that to generate responses, fMRI responses, whole brain responses, potentially for these 50,000 images or 40,000 images, it wouldn't take huge amounts of time. So you would have it in probably 10, 20 minutes, but it's still 10, 20 minutes, whereas we would like you to be able to run your experiments in real time. So we did pre-generate the responses, and the responses, you actually already imported them or accessed them when connecting the Colab to the Google Drive folder that we shared, okay? normally you would, if you decide to use BERG for your own experiments, you will have to generate responses yourself through the code that we shared above. But here we did this for simplicity. Another point which is related to the cross-subject validation issue that one of you asked a few minutes ago. So all of the results from the tutorial, from this part of the tutorial, will be cross-subject validated. So what does this mean? Imagine that you were to run this maximization objective, so you want to find images that maximize responses in one area. Imagine you ran this on a single subject. Like, of course, you will find images that drive responses on that area in a specific subject. But that could be due to the images actually meaningfully driving that area, or it could also be a specific noise-induced or artifact related to the model. So the images end up driving responses, the in silico responses for that area in one subject, but it's not due to underlying signal, it's just due to model noise or whatever. So we want to, instead the tutorial implements in the code is that you will search for the controlling images, the images that drive one area, using in silico ephemeral responses, using all but one subject, and you will get to choose the subjects. You will find this set of images using n-1 subject, and then you will validate these images on the left-out subject. And if you still see that these images, which are validated, cross-validated on the left-out subject, they do end up driving responses on the left-out subject more than, let's say, most of the other images, then you have more confidence that what you're finding is actually related to some underlying neural processes happening, and you also are more confident that if you were to show the same images during an fMRI experiment in vivo, you will find some effects. So cross-subject validation important. And finally, there are two set of ROIs that you can choose among. One is these ROIs which were defined from the natural scenes data set. So you have early visual areas, body selective, face selective, place selective, word selective, and anatomical streams. But you also have this atlas from the HCP. This is also a dropdown which you can expand. And for each area, it gives you the full name and what is the broader swath of cortex that it belongs to, such as primary visual or early visual or premotor, et cetera, okay? One last thing before you start exploring with this, I'd just like to show you how the results will look like and tell you how to interpret them. So let's say we focus first on the neural control part. So the goal again is finding images that maximally drive one area. So eventually, once you run the code, by the way, will take just a few seconds to run, again, because the responses are pre-generator. So it's just about selecting the responses that drive responses, the images that drive responses in one area. And another thing we use for both neural control and relational neural control, they're based on univariate responses, so the average response across all voxels, or in this case, vertices, of one specific area. So say that you wanted to, okay, here I was looking for images that, maximally drove PPA, so pericampal place area. These are other hyperparameters that you can choose from. So there is you have to choose whether you want to use ROIs defined by the NSD or HCP parcellation. Then you decide which ROI you want to use, the image set you want to use, the subject that will be used, the pool of subjects used to find the images, the cross-validated subject on which you evaluate the images, and the number of target images that you want to look for. So eventually the results will look like this. This is the quantitative result. So this is, in gray, is the distribution of all images. On the X is the univariate response. On the Y, the frequency. This is the distribution of all images. The vertical dashed line is the 95th percentile of the distribution. So everything falling on the right of this distribution, you can count it as significantly driving the ROI above chance. And these in red, this is the distribution of the controlling images. So if you see that the images that were selected by the neural control algorithm fall on the right of this dashed line, given that these results are cross-subject validated, it indicates that you found images that actually do drive region of interest above more than chance, okay? And eventually you will visualize these images. So here, unsurprisingly, these images all represent places because we are driving PPA. So this is relational neurocontrol, and for relational neurocontrol, again, here the idea is to find images that either align two areas of your choice, highlighting shared representational content, or disentangle them, highlighting unique representational content. And here, what you will find, so this is the quantitative result. So on the x-axis, you have the univariate response of one area. So in this case, it would be FFA. And the y-axis, the other area, in this case, BPA. And here in gray, these small black gray dots are the responses for all images. And these are the aligning images, images that drive both areas, suppress both areas. These are disentangling images. And these are the chance, this chance. So if you were to select images from chance, you would get this activity. So if anything falls on the right of this, and below of this vertical dashed line or below this vertical dashed line, for example, this cluster here, this is considered significant, okay? And again, this is cross-subject validated. And once you look at the images, again, here it's unsurprising. Okay, for some reason, the images are not plotted. Maybe this cell didn't change, but basically, okay, these images were images that drive FFA and suppress PPA, faces with no scenes. It's like usually zoomed in faces. These are scenes without faces. These, could you have a guess what they might be? Images that drive both areas? Anyone has a guess? Yeah, exactly. Yeah. And these are just usually empty images with nothing much going on, which have neither scenes nor faces. So yeah, you can start now exploring, experimenting. We will be here around. If you have any questions, anything is not clear, please just raise your hand. We'd be happy to come and assist. And now is 5:20. We still have lots of time, almost 1 hour. So I would say you can play around with for 20 to 30 minutes. And then I'd like to have some feedback from all of you or some of you at the end, whether you were finding something interesting or whether there's anything that you would like to share. Why not? Okay, we got 10, 15 minutes left. One thing that I wanted to point out as well is that beyond this tutorial, you can actually use the notebook as a way for doing some exploratory research when it comes to images that maximize specific areas or that align and disentangle other areas. So also feel free to use the notebook for maybe generating hypotheses of how some areas or what they are responsive to. For example, there was someone here, I think they left, but they were looking at auditory cortex and we're finding that, oh, actually there are some stimuli that significantly drive, visual stimuli that significantly drive auditory cortex. So it could also be, yeah, hypotheses generative. You could use it for hypothesis generation. But this being said, is there any of you that found something interesting or that you would like to share? If you don't want to share it in front of all of the group, we can also I don't know, come there and you just tell us what you found. Is there anything unexpected or else that you would like to share? If not, it's also cool. Yeah, in general, so as we were mentioning also earlier, we're really happy to receive comments, input for improvement regarding not only this tutorial, but specifically BERG, the toolbox. So if there are things which do not work, you find errors, you find functionalities that you would like to be implemented, we're really open for feedback. But especially if you do have models that you would like to contribute to the resource, we would be more than happy to integrate your models. Domenic did a really nice job in making the model integration process as easy as possible for you, so it shouldn't take too much time to add your own models to a GitHub pull request. You can always get in touch with us, of course, if you wish to do so. And the benefits of this is that even maybe for yourself in your own lab, if you integrate your model to BERG, then you can use it to generate responses with this one-liner of code, which is maybe much easier than every time having to run your whole functions. Another benefit is that you can share it with basically everyone. So everyone will be part of this list of models which are available in BERG. So if you want to make your model more usable by others without having to look into your code base and using some standardized code, this also makes it possible. So yeah, if you have models that you want to contribute, we'd be happy to have them. Again, we started doing this and adding models ourselves, but if the toolbox is to grow and to integrate more models, we also need your help for this, because we obviously have also our own research to do, and we cannot continuously add new models ourselves. So we're looking forward to your contributions. Cool. I guess we will be around for another 10-15 minutes if some of you are still doing some work and you have further questions. And if not, obviously you can leave at any time. And thanks for joining.